Okay, well, I'd like to start off and thank everyone for coming. This is a combination of learning about some interesting science that is very applicable to, I think, in many ways, the biotechnology and cultural events, but also is in many ways a very personal conversation. Uh, so we're going to talk about contributions to cryobiology by the late Stanley Leibo. And he was someone that I knew as another faculty member at the University of New Orleans. And he passed away almost a year ago from cancer. And I knew him largely as the older guy who did some research with freezing stuff, at least for about the first 3 or 4 years that I was at UNO. And again, being busy, didn't spend a lot of time interacting with him per se, because he actually was very busy traveling and conducting a research program. About the last 3 years I was there, he actually started teaching some of the basic biology classes. And that's an opportunity where he's like, I hear you know a little bit about technology, so maybe you can help me get some stuff set up. And from then, we really started interacting more. And what was always really amazing about him was his enthusiasm for science and his enthusiasm for really interacting with people and talking about stuff. And so he always had a number of great stories to talk about in terms of, again, some of the very classical big figures in biology, not just in his field of cryobiology. So I felt that the best way that I could honor him in passing would be, one, to learn a little bit more about his field and his research. And that is something that, again, is something I've started doing, and I'll share a decent amount of that. But then also to honor him by conveying what his work and that field does to, again, a more general audience. So thank you for helping me with that second goal, and I hope you enjoy it. He did his graduate work at Princeton University, and then also moved on to Oak Ridge. And this is a picture of him from the obituary that was on the International Society for Embryo Transfer. And again, showing him hard at work, involved in a training program that he constantly put on throughout his lifetime. Now, what I would like to, during the last part of his professional career in terms of appointment was at the University of New Orleans from 1999 to 2014, where he mentored graduate students, taught classes, and was also involved with the Ottoman Institute, involved in some research looking at species preservation. So I'm going to start and mostly talk about cryobiology. And again, this is a definition that comes from the Society for Cryobiology. The definition being, again, cryo meaning something cold, and then biology related to biological organisms. And, you know, the professional aspect of the field really revolves around trying to preserve biological specimens with the integrity of keeping them viable and using them for some biological purpose. Some of these examples that they mention are long-term storage, and then cryosurgery, stabilization of pharmaceuticals, study of cold adaptation in plants and animals. So again, you've probably heard of these fish and other biological organisms that live in say, the Arctic or the Antarctic oceans. But the large majority of the focus of Dr. Leibo's career actually had to do with preserving cellular components of organisms in order to then revive them. So in terms of cryobiology, a somewhat specialized area within that. And some of these applications would involve the preservation of gametes, again, either sperm, spermatozoa, oocytes, or the ova. Preservation of embryos, so again, after fertilization, either the 1 cell to 8 cell to the blastocyst stage, where there's, again, maybe a couple hundred cells involved. And then, and I have an asterisk here, also the study of cryonics, where again, people or pets preserve their bodies through freezing at very cold temperatures. And again, this is something that is I put an asterisk because many hardcore cryobiologists would not include cryonics in this type of field, in the sense that we don't really know that we can, or there's just a hope that those can be revived to become then a viable intact organism. One of the most well-known applications of cryobiology is related to, or is one component of what's known as in vitro fertilization. And so the Nobel Prize was awarded to Dr. Edwards in 2010 for the development of in vitro fertilization. And this is a process by which gametes, eggs, can be extracted from a female, put in vitro inside a little dish, which is what in vitro means outside a cellular system in a test tube, and then exposed to sperm, one of which would fertilize it, begin the embryogenesis process, and then ultimately re-implant that back into that woman or another surrogate's uterus for then ultimate development into a new person. Or again, when applied to, say, other mammals or amphibians, lizards, would lead to a new individual. So one aspect, of course, that makes in vitro fertilization most powerful, like you can do this without freezing anything, it's something very direct, but in terms of its wide-scale applicability in culture and in livestock, then it's something where being able to freeze stuff so you can be able to more conveniently implant it and have surrogates, that really is what makes it a much more feasible technology. Now, in terms of the numbers of situations in which this has occurred, there have been millions of cattle that have actually been, had embryos frozen and then reimplanted to lead to a live birth. Again, this is one of these things where if you have cattle stock, it's much easier to transport large numbers of either very specialized breeding strains or even transgenic cattle by airplane, say, across continents, by being able to just transport frozen eggs as compared to whole cattle. And then also, you sometimes don't know how useful a particular breed or breeder an animal is until maybe a little bit after the fact. So then you can go back and, again, have more copies of it later. Pretty much all the common experimental animals that you find in biomedical research, the mouse, the macaque, hamsters, these have all repeatedly and consistently been shown to be something that you can preserve. And again, for science, it's very useful to, if you develop strains or breeds or transgenic animals, to have them frozen away so that either you can ship them to other researchers or that you can go back and analyze them maybe in a new method or new types of experiments after you've had the wisdom of time and other experiments, look back and say, oh, I wish we had done that. There have been tens of thousands of people that have been born via in vitro fertilization and also with cryopreservation of the embryos. And of course, one of the things that I think is going to be, and one of the contributions that Dr. Leibo was making near the end of his career, was genome banks. That for a lot of species that are, their wild populations are disappearing from the planet, that if we have the opportunity, if we can get ourselves around to finding a way to preserve the habitats for these animals, if we have them frozen away as embryos, or again as their sperm and eggs, that's something that we could then revive species that are endangered. And again, there's a question in the audience about, you know, the freezing of maybe other types of cells. I was trying to focus here mostly on those that are involved with Leibo's work, but yes, the preservation of many cell types is again, something ongoing, very important for biotechnology, including things like umbilical stem cells, which, depending on how you view them, those are very useful for potentially replacement cells for that individual, or to be used for other purposes like stem cell differentiation. Now, this is something that does hit the popular culture in many cases. Probably all of you that are stateside, and maybe some of you in other countries as well, heard about Nadia Sulman, also known as Octomom. And again, she's someone who received a certain amount of celebrity slash what I would also call slash notoriety for being a woman who was really only the second person at the time to have given birth to eight human offspring at one time. There have been others since that time. And what was very interesting about this, and I think something that again permeates the popular culture, is that a lot of people felt she, there's a lot of moral judgment involved in how people perceived and discussed this particular situation. I think, again, one thing to keep in mind is that while she made the request to use the remainder of frozen embryos that she had from earlier in her life, the actual ultimate decision for how many to implant was made by the doctor. And he actually ultimately, from what I read, lost his medical license for having implanted so many at one time. So in terms of the scientific literature, cryobiology is a growing field. Again, cryobiology is something that really hasn't even really reached about 100 years of age in terms of the very first early publications. But it's a growing field in terms of basic research that I just performed earlier this morning, a PubMed search using the key terms that you see here. And these are the numbers of papers that were published, again, which could include review articles in the year 2014. And there's a video of Dr. Leibo giving a talk at Cryo 2013 in which he talks about, he actually shows a whole graph from the beginning, that in essence, there's an exponential growth in the number of publications related to cryobiology. So it's a field that's growing and has, I think, a very rosy future. Now, to introduce you to a little bit more about what cryobiology really is about, is that I think one way to sum it up, and what I've seen from several of the people in the field, is that cryobiology is really about the study of trying to avoid ice damaging tissues. And so this is an image of freezer burned hamburger, where if you put meats or any number of items in your freezer, and they're allowed to sit there for a pretty long period of time, and they probably also go through some freeze-thaw cycles. Again, not a freezer that just stays frozen, but these ones that are more common that undergo, again, thaw cycles so they don't accumulate ice. Then you also don't wrap them up and preserve them. Then this is the type of hamburger meat that you get out of time. And this is something that can affect the palatability, the flavor, and in many ways the texture of the meat. So I think this is something has anybody ever done this where you're maybe really, really hungry and you look back in that freezer and say, maybe, but maybe not. And again, this is just in terms of food that you might eat. And again, think of the potential implications when it comes to intact cells. And that, again, if you're preserving embryos to be a future child, that this is not the type of thing you'd like to have happen to it. So what is the chemistry involved? And I think in terms of understanding, ice, one of the really interesting features of ice, and I'm not going to go too much into the chemistry, is that when you get it really cold, the molecules like to line up and interact with each other to form crystals. And again, this is an image from an online biology textbook, boundless.com. And it shows water molecules forming a kind of crystalline lattice that, again, you'll notice the spacing of them is very regular. And one of the consequences of that spacing is that ice is less dense than liquid water. And so that's why the ice flow is the picture right adjacent, is that again, when you think about ice, it's always something that's floating on top of water. And so that it's important to recognize that that's the lower density of it. But also, again, if you have water inside a cell, then as those crystals form during the freezing process, then they will also expand the tissue, interact with the membranes to actually take up more space. So if you've ever tried to cram a suitcase or ever tried to fill a water balloon up with too much water, this is the type of process that occurs. And this is something that's this is basically what kills living tissue when you freeze it. A lot of you are probably familiar with one of the ways that we overcome this problem. Again, this is a problem for cars, that in order to maintain engines from getting too hot, we have radiator fluid in them. And that radiator fluid helps distribute the heat, so it's not all just connected to the metal. But if you just had fluid running around your car, just plain water, which would distribute heat nicely, if your car were to get to freezing temperatures, then any ice that formed in the tubes, in the pipes, would expand and then crack the pipes, or else rupture the rubber. And again, once you leak that fluid out, then it's no good to you. So everyone's very familiar with this idea of antifreeze. And so the molecules that you'll find, propylene glycol, the molecular structure that's on the bottom left, again, a couple carbons, then a couple oxygens and hydroxyls, is what you find in most modern antifreezes. It's less lethal than the molecules to the right, ethylene glycol, which is something that if you were to drink it, has a very bad crystal formation in your kidneys, and will shut those down and cause you to die from not being able to filter out the waste. And then things like ethanol and methanol, glycerol, other common biological organic molecules also work to, again, keep ice from becoming a crystal as you go below 0 degrees Celsius. Again, another molecule I have there on the bottom right, it's not used as an antifreeze. It's actually rather, it's somewhat toxic to biological organisms at high doses, is dimethyl sulfoxide. Again, that's not something you'll find in consumer products, but it is something that's been used in cryopreservation techniques. So I mention it here. And again, the way these types of molecules work is by including them in a solution of water molecules. Then they interfere with those bonds between the water molecules, between the hydrogens and oxygens, in a way that you can never get a proper nucleation of crystals, and thus the process is less likely to damage cells. But nonetheless, one thing to keep in mind is that any time you, but what's important to recognize is that just that alone, having solutes in solution is not enough to keep the cells from being damaged. And so one thing that everyone's pretty familiar with is that an overexposure to very cold temperatures can lead to frostbite. And so that's something that can lead to gangrene and the ultimate amputation of limbs. Once you've interrupted that blood flow, you can't get oxygen to the tissues, and they will die. It's also very painful. Again, this is just an example of Jack Nicholson in The Shining, and that this process, that even though the scientific community had these types of antifreezes, that in most cases, they were still finding that for a lot of these, for embryos in particular, they were still sensitive to the freezing process. That even just mixing the two together wasn't a very good guarantee that you could then thaw the cells and get them to actually be viable, implantable, lead to new organisms. And this was particularly true in mammals. And so again, this is the main scientific contribution that Dr. Leibo made to the field, is that in combination with the visiting Dr. Whittingham and his graduate advisor, Peter Mazur, in 1972, they published the ability to actually for the very first time ever, gotten a mammalian, the mouse, to freeze the embryos and then lead to successful pregnancies in multiple surrogates. So I'm going to walk slowly through this graph so that, again, this is really the only graph I want to show during the talk, and so I want to take a little bit of time to go through it. That what you actually see on the x-axis on the bottom is the cooling rate. And so going from left to right, again, the rate of cooling is the number of degrees per minute at which the embryos were frozen. And so going from left to right, lowest to highest. And that's what you have for both graphs. And on the y-axis, you have the percentage survival of the embryos. And in this case, percent survival is defined by 2 or 4 cell stage zygotes going from that stage to actually under the microscope seeing them becoming, going through the blastocyst progression. So showing that they progress to the next phases of embryogenesis. And what you'll see in both graphs is that the slower the cooling rate, then the higher the viability. And that no matter what cell type you have, if you try and cool something down too quickly, then nothing survives. And so, again, I've read up on quite a bit of literature at this point, and Peter Mazur gives a very nice summary of this at his Cryo 2013 talk, that just because you've added antifreeze molecules to your solution, there are 2 things that a slow rate helps you with. Is that one, it's not just that you have water not becoming crystals, it's also the rate at which you dehydrate the cell. And so if you are slowly dehydrating the cells, then whatever sort of osmotic pressure or whatever sort of cell function and structure you have, the slower you pull the water away, the better. And that really more importantly is that as you freeze stuff, you're still forming microcrystals. And so those microcrystals have the capacity to create cellular damage. So if you're doing it more slowly in the presence of an antifreeze, it specifically inhibits those microcrystals from forming and thus causing damage to the cell. And what I have here at the bottom is that, again, if I interpreted the table correctly, that 65% of the 501 embryos actually successfully went through progression in the mice, and that 48% of the implanted embryos that were allowed to go to full term, there were some mice that were sacrificed in the middle in order to look at the progression, so they weren't allowed to go to full term, that 40% of the implanted embryos allowed to go to full term were born. And so that ended up making the cover of Science magazine for that publication. And then, in addition, was certainly a great cause for celebration. Now, so this methodology of what's called slow cooling is still the primary and most popular method for the vast majority of mammalian embryo freezing. And then Dr. Leibo has a history, a large number of publications that talk about this methodology for a variety of cells, including spermatozoa, sperm, oocytes, ova, as well as other species embryos. There's another technology called vitrification, which I didn't really find any particularly helpful images to explain the process, but vitrification is a process by using other chemicals where you can very rapidly cool cells down in the presence of antifreezes, and you actually get a glass-like, again, an amorphous phase structure of water, such that, at least in human embryos, you have actually a higher survival rate than you do with the slow freezing. But again, it's not a technique that's being abandoned for human embryo freezing because it's actually a lot easier to handle large numbers of samples, and it's a little bit less sensitive to, again, how technically specifically you're doing the protocol. So again, this paper is basically credited with the explosion of frozen embryo transfers that, again, you find very widespread in livestock as well as in scientific labs. And again, they overcame something that was a very specific technical challenge that even though people knew you had antifreeze technologies, the ability to get a high rate of survival of the embryos was, of course, a milestone in the field. To summarize Dr. Leibo's contributions from that point on, looking in World Science, he had a total of 164 publications. Many of those are reviews, but if you actually look at the variety of science that he performed over his lifetime, there are a large number of species involved, including mice, fruit flies, rabbits, white-tailed deer, and rhesus monkey, the macaque, and the cheetah. And again, the cheetah publication, I actually saw the graduate student present some work related to that so the ability to freeze sperm and embryos and eggs from a wide variety of species is actually very challenging, because one of the main differentiating things about new species is how their gametes interact. And that's something that can evolve very rapidly. And that they all develop very specialized interactions that mean and has the consequence that for these freezing technologies, one protocol that works for one organism may or may not work for another. And so interestingly, one of the challenges that is seen for cats is that the sperm almost have a very low survival rate with whatever sort of freezing technique you have. So that was the work of a graduate student where I saw the talk, where trying to find ways to modify the protocol to allow a higher survival rate is something that, again, people are very interested in. So when it comes to the cryopreservation of rare or threatened species, making sure you work through the technologies, finding ways that this is reliable to actually save them and preserve their genomes for maybe decades until we can deal with habitat loss. Fine-tuning these techniques are very important. And that's one reason why, well, actually, let me show that later. Now, during his time, he was not just an academic, he actually was a director of animal laboratories, both at University of Guelph and a company, Rio Vista International, down in San Antonio, Texas. He does have 2 patents to his name related to embryo transfer methods. Later, he was also awarded several, again, outstanding professional awards. This is just a highlight of a few. The Pioneer Award from the International Embryo Transfer Society. He was also awarded an honorary membership and given an award from the Spallanzani Lecturer for the Society for Spermatology. He was awarded, or he was inducted as a fellow of the Society for Cryobiology, recognizing his exemplary contributions to the discipline of cryobiology and service to the society. And I think that's something I want to talk about. I'm going to show some of what his contributions were to that. And as well as being an honorary lifetime member of the American Embryo Transfer Association. I'm just going to highlight a couple of his professional society appointments. He was the president in the 1980s, both for the Society for Cryobiology as well as the International Embryo Transfer Society. And one of the interesting dilemmas that came during this time was the argument between whether cryobiology should or should not really accept or recognize cryonics. So I mentioned that cryonics is the attempt to preserve humans, or in some cases pets, using freezing technologies in order to then hope that future technologies will allow those to be revived. And in fact, this is something where you see some degree of ridicule in the popular press or in entertainment. If you've ever watched Futurama, where they have the little heads in floating jars. Again, I hate to spoil this, but if you haven't seen it by now, you probably are not going to, is that the movie Vanilla Sky, both the original as well as the Tom Cruise, the main plot twist there is that the main character has been cryonically frozen and then is ultimately revived. And so, again, I think a lot of people credit him, even the cryonicists, that a lot of people were very much against this, and this got to a lot of heated voices, but it's generally regarded that Dr. Leibo guided that argument and was a cooler head in the overall conversation. In terms of being a mentor to the field, every year from the beginning and the founding, there's a workshop in the cryopreservation of mouse germplasm, which is sponsored by the Jackson Labs in Maine. For those of you unfamiliar with the mouse research field, the Jackson Labs is an invaluable resource for people doing research with mice. They maintain a variety of mouse strains. They maintain very specialized transgenic versions. It's a repository that allows the field to not have to have multiple sites and multiple people trying to maintain and create mouse stocks. And so very invaluable. And he would every year provide a workshop related to mouse cryopreservation. And so from what I've read, there are probably hundreds of students have gone through that. So there are probably lots of hands out there that have been guided originally by Dr. Leibo's experience. He was also the dissertation supervisor for a total of 12 students. Again, this is information I got from his curriculum vitae, which, let me mention, was kindly provided by the University of New Orleans biology department after his passing. That's one thing that's actually kind of interesting about Dr. Leibo, is that since he's been around the field for so long, there actually aren't a lot of easy web resources to track down some of the non-publication aspects of his life. I'm going to throw a link in here at the end, but one of his last grad students is actually a research faculty still doing cryopreservation, and she actually put a YouTube memorial together as well. So I'll post that link in at the end. His contributions did not end with the research. He was, again, one of really the primary roles of academic faculty in the research area is to maintain the communication and to both nurture journals and be a member of editorial boards, but also to review lots of manuscripts. And so he was heavily engaged and involved in being a editorial board member, a regular reviewer, but pretty much all the major journals involved in cryobiology. And again, one thing you'll notice here is that a lot of these were to the present. And so I think one thing that Dr. Leibo, when he made a commitment to doing something, he really was going to stick to it. And I would like to just close here with just a personal reflection on this, is that one of the motivating reasons for wanting to not just honor him personally, but to give a presentation, let other people know about his contributions to cryobiology, is that there are a lot of people that probably are colleagues of yours that when you're busy, you don't necessarily focus on trying to get to know them personally and to spend that time trying to very specifically understand not just what they've done in the field or what they are, but really the stories that they can share about what has gone on in the field and their perspectives of what science used to be like. It's interesting in that the reason I started interacting with him more was that he wasn't very familiar with clickers in the classroom and trying to get Blackboard to behave and those types of things. And so I have to say that it's something I am ultimately very thankful for in terms of being one of those go-to people with helping with technology, although many times it can be very frustrating, that it gave me the opportunity to actually get to know someone who is both a very a luminary and a pioneer in his field, and also someone who is just an extremely, extremely nice man, and very generous with his time and very generous with his expertise to the field. So I'm going to end. I'm going to throw a link in here from his, as I mentioned, Jennifer Barfield, who was his last, as far as I know, UNO PhD student, and is currently a researcher. So I'll go ahead and click that, and when you're done, just let me know in local chat that you're done, and then I'll throw in one more link so you can have that for posterity. Okay, I'm going to go ahead and again, thank you all for attending and listening. I'm going to answer the question I saw from Moonsnail, which is, does cryopreservation make gene manipulation easier in the long run? For example, freezing cells at different stages of growth post-insertion. And again, the cryopreservation of a variety of samples and research, again, even just including E. coli, bacteria, and yeast, creates a powerful way to examine biology and to, again, perform an experiment. If you do get useful results out of it, then you can always go back and take your other similar copies. It really provides a very nice safety net. One example, there's been a 30-year-long experiment with an E. coli culture in which it's been continuously growing and then branched out under different conditions, showing specific evolution over different times. And what they've consistently done is frozen away samples. So you can actually compare the evolution of E. coli 30 years later and actually go back and compare it to the original culture and sequence the genome, look at the specific mutations. And so that type of preservation power does really complement the types of experiments you can do. It's really like going backwards through time. And so that's a very good observation and question. RF asked the question, cryonics is the modern day waking up Rip Van Winkle or return after 100 years in fairyland scenarios. Maybe that's why people get upset. I think that's a part of it. I think there are plenty of people who feel when you disturb the natural order of things. Again, biological systems are all terminal. There's the design, it's not a design flaw to die. And so I think a lot of people get upset when you break the natural part of it. And then the other thing to keep in mind is that just in terms of cultural disparities, that rich people can afford something like that, whereas it's not so affordable for poor people. And so I think that that's something that very rationally is like some people are getting benefit that others can't take advantage of. Berwin asks an interesting question. Evolution must have found mechanisms to all allow single cells to survive snowball Earth. This seems relevant to cryobiology. Is this an area of research? What were the mechanisms for cyanobacteria? Again, what Berwin references is that approximately 1.3 billion years ago, there was a cooling off of the Earth, a lack of CO2 in the atmosphere that led to basically snowball Earth, meaning that the glaciers went all the way to the equator. Luckily, some volcanoes erupted and we warmed back up. In terms of developing mechanisms, again, there are lots. There's a growing interest in biology research for extremophiles, organisms that can or are adapted to extreme environments, not only for understanding and having useful applications in biology, but also in terms of, is there life on other planets? Again, one example, if you've heard of PCR, which is the ability to rapidly amplify lots and lots of DNA, that's actually based on an enzyme from Thermus aquaticus, which is a deep sea vent organism. So its enzymes are adapted for like really high temperatures. In terms of whether someone's gone back and looked specifically at the adaptations from 2.4 some billion years ago, that's not really clear to me. But there are lots of adaptations that people found. There are lots of bacteria that live in the Antarctic ice. I remember reading several articles about that. Again, they can live in low sunlight. They can live in water. They have these. Again, they all develop, and they all have the capacity to express a lot of times glycol type or glycerol related compounds allow them to not get frozen and have ice crystals. So that's kind of a long answer, but yeah, I can't answer it more specifically, but there definitely is a rich body of research looking at that type of thing. Valley Wave Rider asks, what's the largest, most complex organism that has been cryogenically frozen and then revived? I don't know. I will say that there is an attempt to revive the woolly mammoth from frozen, ones that have naturally been frozen from 10,000 years ago, to take cells from it. Again, not revive the whole organism, but take cells from those and then see if you can implant them in an elephant and get that to survive. Because that would be, I think, kind of awesome. So I don't know. I don't know about that. Now, in terms of whole organisms being frozen and revived, where it's actually been completely submerged and frozen, Dr. Leibo talks about some rats that a woman whose name I'm blanking on was able and demonstrated in videos from like 70 years ago that she could completely freeze them and then revive them in a way that's, again, they were frozen on the outside, but they weren't completely frozen into ice on the inside. And so whatever sort of preservative she used didn't have to necessarily permeate the whole organism. Uh, so that's, I think frogs have been, and rats, but I'm not aware of anything larger than that. A deep frozen organism revived. Yeah, I don't know. I just don't know. Again, this is in no way my field, and so I largely focus on the small cell stuff related to Dr. Leibo's work. Well, tardigrades and some of these other mite organisms and water bears, they have a dehydration mechanism. So yeah, there are, I guess if we think about cryopreservation, including just straight dehydration, there are lots of examples of organisms that do that. Deloid rotifers do that as well. Thank you, Chantal. Thank you for giving me the opportunity to do this. Um, Thank you all again for coming and listening. And again, think about those people that you can learn from and take the opportunity.